Today, our discussion is on Mendel's experiment. In 1860s, Gregor Mendel developed the model of heredity that now bears his name by experiments on various characteristics of pea plants, such as height, tall versus short, seed color, yellow versus green, seed coat, smooth versus wrinkled, etc. The following explanation uses the tall versus short trait. The other traits Mendel studied can be substituted for tall and short. He studied this by crossing two pea plants with alternative characteristics by artificial pollination. Let us look at his experiment in a detailed manner. Mendel started out with plants that bred true. What do we mean by bred true? When both the alleles are homozygous, that is, either both alleles are dominant or both the alleles are recessive. The particular organism is bred true for the respective characteristic. In other words, when tall plants were self-pollinated, or cross-pollinated with others like them, plants in following generations were all tall. When the short plants were self-pollinated, or cross-pollinated with others like them, the plants in following generations were all short. By assuming that tallness is dominant over short, let us denote the tall parent plant in uppercase T and a short parent plant using lowercase t. By looking at the experiment results, Mendel found that if true breeding tall plants are crossed with true breeding short plants, all the next generation of plants, called F1, are all tall. Next, he showed that self-pollinated F1 generation plants, or cross-pollinated with other F1 plants, produce an F2 generation with 3/4 of the plants tall and 1/4 short. In this figure, look at the first row of the figure. I am showing you how two plants, one is tall bred true and the other one is short bred true, can be pollinated. I have shown the resulting offsprings on the right side, where we can see that all the offsprings are tall now. This generation is called F1 generation. Now let us look at the bottom row. The bottom row shows that two plants from generation F1 are pollinated to obtain the offsprings in generation F2. We can clearly see that 75% of the plants in generation F2 are tall while 25% are short. In the generation F2, Mendel furthermore found that 1/4 of the F2 generation are short plants, which produce only short plants in the F3 generation, if they are self-pollinated. These F2 plants breed true. 1/4 of the F2 generation, 1/3 of the tall plants, are tall plants that produce only tall plants in the F3 generation, if they are self-pollinated. These tall F2 plants breed true. 1/2 of the F2 generation, 2/3 of the tall plants, are tall plants that produce 1/4 short plants and 3/4 tall plants in the next generation, which is F3, if they are self-pollinated. This is the same proportion of tall to short that F1 plants produce. Now, based on what we have discussed so far, try to answer the following question. When Mendel put pollen from tall plants into the flowers of short plants, the seeds produced an F1 generation with all tall plants. But the short trait was not lost. How did Mendel demonstrate this? The correct answer for the previous question is: he showed that self-pollinating F1 plants produced 1/4 short and 3/4 tall plants. Here are some key points that we need to consider. Transmission of each trait from one generation to the next is carried out by discrete units, which we now call genes. The gene for each trait comes in pairs, one from each parent. This deduction was confirmed by studying chromosomes, which are visible under the microscope during cell division. Chromosomes come in pairs and carry the genes, which themselves are invisible under the microscope. Now, can you try this question? Here is another question. Similarly, try to answer the remaining questions as well. Here are the correct answers for the questions. Genes for a trait from the two parents remain separate. They do not blend. This explains the fact that short plants and plants with wrinkled seeds disappear in F1 but reappear in F2. This known as the law of segregation. When Mendel measured two or more traits, for an example, height and color, in an experiment he found that each trait was transmitted independently. For example, tall or short plants can have smooth or wrinkled seeds. This known as the law of independent assortment. There is a technical way of identifying the genotype or phenotype compositions from one generation to the other. Before we discuss it, let's see whether you can answer the following question. Let us consider the coat colors in a guinea pig. Now, some of you may wonder what is a guinea pig. It is a tailless South American rodent, rat, of the cavy family. Now again the question. Let us cross a homozygous black guinea pig with a homozygous white guinea pig. If black is the dominant allele, calculate the black to white ratio in the F2 generation. There is an easy way to determine the genotype and phenotype compositions. The Punnett square, a square diagram, allows us to predict the genotypes of a particular cross or breeding experiment. In other words, this is a simple graphical way of discovering all of the potential combinations of genotypes that can occur in children, given the genotypes of their parents. As we learned earlier, a dominant allele is represented by an uppercase letter while a recessive allele is represented by a lowercase letter. Let the skin color trait of an organism has two allelic forms, an uppercase T is to denote dominant black and a lowercase t is to denote the recessive white color. Whenever the genotype of an organism is 2 uppercase Ts, the skin color of the organism will be black. This is called homozygous dominant genotype. Whenever the genotype of an organism is uppercase T and lowercase t, the skin color of the organism will be black. This is called heterozygous genotype. Still, the color is black since there is a dominant allele in the pair of alleles. The only instance where the color is white will be whenever the genotype is 2 lowercase ts, which is a homozygous recessive genotype. Now, it is evident that whenever the genotype is known, we can determine the phenotype, physical appearance. But the reverse is not true, in general. Let us see how to represent a cross between two parents using a Punnett square. We need to stick with only one genotype for the moment. In this figure, I have shown you the Punnett square for two parents, trees, where we need to determine the leaf color of offsprings. One parent with heterozygous alleles, indicated by an uppercase Y and a lowercase y. Look at the letters appear in front of each row, outside the square. The other parent with homozygous alleles, indicated by two lowercase ys. Look at the letters appear on top of each column, outside the square. Based on the alleles of the parent trees, the inner content of the square is filled. Cell in first row and first column gets the value upper Y and lower Y. Cell in first row and second column gets the value upper Y and lower Y. Cell in second row and first column gets 2 lower ys. Cell in second row and second column gets 2 lower ys. Now based on the genotypes of the offsprings, we can determine their phenotype. For this example, we see that one half of the offsprings will have green leaf color, and the other half will have a yellow leaf color. Let's consider another example. Here we consider the color trait of a flower, where purple is dominant and white is recessive. Graphs shows 2 generations when the F0 generation is from 2 homozygous parents with dominant and recessive genotypes. By trying to answer these 2 questions, it confirms that whenever phenotype is known, it is difficult to determine the genotype. Coming up are few example exercises that you could try to answer. I will be discussing the answers for these questions when we meet for an online discussion next week.